welcome to Biology Paper 2, the video on inheritance. So, to start off with, we need to look at sexual and asexual reproduction and the key differences. So, with sexual reproduction, it involves the fusion of gametes, which are the sex cells. So, in animals, that's thinking about the fusion of a sperm cell and an egg cell, and in flowering plants, it's the fusion of pollen and an egg cell. So, with sexual reproduction, there's a mixing of genetic information, which creates variation. And the gametes that are necessary for sexual reproduction are produced via the process of meiosis. On the other hand then, so we've got asexual reproduction, which is where there's one parent, there's no gamete fusion, and there's no mixing of genetic information. So what that means is that the offspring that are produced are identical to the parent, so they are called clones, and the way in which the clones are produced is via mitosis, which we looked at in paper 1. So, just as a bit of a recap then, because we need to kind of be aware of both of them still, mitosis versus meiosis. So, with mitosis, we've got two genetically identical daughter cells being produced, and those daughter cells are diploid because they've got the same number of chromosomes as the parent cell. In meiosis, we've got 4 non-identical daughter cells being produced, and those cells are going to be the gametes, which are the sex cells, and during the way in which they're produced, the number of chromosomes is halved, so those cells we can call them haploid. Okay, so 4 non-identical haploid cells are produced. So, just as a kind of recap then of meiosis, you start with your parent cell. The DNA is duplicated, and then we undergo one initial division, which splits it down into 2 cells, okay, each containing 46 chromosomes, and then there's a second round of division where those 2 cells split into 4 cells, which each contain 23 chromosomes. So those are going to be haploid because they've got half the genetic material of the initial cell, and they are going to be the sex cells. They're going to be gametes that are being produced. Now, it's important to bear in mind gametes, when they join at fertilization, they will restore the full number of chromosomes. So obviously a sperm cell would have, if it was a human, 23 chromosomes, an egg cell would have 23. When they fuse together, that's going to give 46, which is the total number. The new cell that's then produced, so when that sperm and egg are fused together, will actually divide by mitosis as the embryo sort of develops, and the cells will differentiate into all the different types of cells. So, moving on then to the genome. What a genome is it's the entire genetic material of a living organism. There's something that we need to know about called the Human Genome Project, which is when we've managed to actually sequence all of the genome of a human. So we know kind of all the different bits of DNA that build up the human genome. This has been really useful in terms of advances in science because it's enabled us to link certain genes to diseases. It's enabled us to understand treatments a lot better, and it's also enabled us to trace human migration patterns from kind of like a historical point of view. DNA in the genome then. We need to be aware of the basic structure of DNA. So it's got a phosphate backbone that forms kind of a double helix because there are like 2 strands obviously that kind of coil around. And it's also made up of 4 different bases. So we've got adenine, which is the one that will bind with thymine, and then cytosine, which will bind with guanine. Okay, so 4 bases: adenine, thymine, cytosine, guanine, and they pair up A to T, C to G to actually form that proper helical structure that DNA is. Another quick recap then. Obviously, inside cells, we've got a nucleus. The nucleus contains the chromosomes. Chromosomes are made up of DNA, and short sections of DNA are what we call genes. So the reason genes are really important then is because they're small sections of DNA that will code for a specific protein. So every 3 DNA bases in a gene will code for 1 amino acid. Those amino acids will join together to form a specific protein, which would be, essential for a whole variety of different things depending on what that protein is. But for instance, it could be forming enzymes. The next bit then that's part of inheritance is kind of the genetic side of things. So with this one, we've got a lot of keywords that we need to bear in mind. We've spoke about some of these already, but just as a quick recap, we've got gametes, which are the sex cells, chromosomes, which are made up of kind of DNA coiled together, found inside nucleuses, genes, which are short sections of DNA that code for proteins, Alleles, which are an alternative form of gene. Dominant, which is the type of allele that we represent with a capital letter, and only one of those needs to be present for it to be expressed. Recessive alleles are ones with a lowercase letter. They need two of those present to be expressed. Homozygous, so homozygous genotypes is when we've got two of the same letter, so it could be two capitals representing two of the same allele, two lowercase representing two of the same. Heterozygous is when we've got one of each allele, so an uppercase and a lowercase letter to represent those. Genotype is thinking about the two alleles that kind of are present. Phenotype is the one that's relating to the physical appearance of the genotype. So genotype is actually, what alleles are present, what letters have you got. Phenotype is the physical appearance of it, so do you have blue eyes or brown eyes, for instance. So, when it comes to inheritance, some characteristics are controlled by a single gene, so for instance fur colour in mice, which we're going to look at in a second, but most are actually a result of multiple genes interacting, so it's quite a complex thing. So, for higher tier people then, we need to know about genetic crosses. So I'm just going to quickly go through one with you just to recap. So we're going to use mice as an example. So if we say, for instance, with mice and their fur, if we'll say that black fur is dominant, white fur is recessive, I want to know the probability of kind of white or black fur offspring if the parents are both heterozygous. So if they're heterozygous, they're going to be containing one of each allele, so they're going to have a dominant and a recessive allele. For ease, we're going to say today that black fur is going to be a capital B and white fur is going to be a lowercase b. So because they're heterozygous, the parents are both going to be big B, little b. Okay, so we put them into our cross. We then need to work out the offspring. So here we're going to have capital B, lowercase, capital B, lowercase b, lowercase b. So we then want to know what percentage are going to be white furred versus black furred. Because black fur is dominant, we're going to end up with this one here, which is homozygous dominant, having black fur. These two, which are heterozygous, are going to also have black fur. So that's going to give us a 75% chance of black fur. Okay, and then the one that's going to be white furred, because it's homozygous recessive, is going to be this one here, which is 25% white fur, because it's got both of those recessive alleles, both of those lowercase ones. Okay, so 75% black fur, 25% white fur, or, you could say 3 in 4 will be black furred, 1 in 4 will be white furred. Okay, final little bit for inheritance. Inherited disorders, we've got two we need to be aware of primarily. Polydactyly, which is when there's extra fingers or toes. This one is controlled by a dominant allele, so a capital letter will be representing that. Only one needs to be present. Cystic fibrosis then is a disorder of cell membranes, and this one's caused by a recessive allele, so it's got to have two of those alleles present, two lowercase letters to represent it. Final bit, sex determination. Chromosome pair 23 is the one that determines the sex of a human. 1 to 22 are just kind of determining all kinds of other things to do with characteristics, but pair 23 determines sex. XX is female, XY is male, and if you were to do a genetic cross of this, you would see that there's a 50% chance of a male or a female offspring. Okay, so it's always 50-50. And that can be quite easily demonstrated by a genetic cross.